HIF2 alpha inhibits glutamate clustering in mitochondria to sustain growth of clear cell renal cell carcinoma. Let's meet the starting lineup. We have glutamine, which is a nutrient that cells use for energy, growth, and cell survival. Glutaminase, or GLS, a protein that helps break down glutamine so it can be used as fuel. Blocking GLS can make it harder for cancer cells to grow. HIF2 alpha, a protein that helps cells adjust when oxygen and nutrients are low, and CRCC, aka clear cell renal cell carcinoma, the most common type of kidney cancer. Introduction. Glutamine is an important nutrient that many cancer cells use for energy and growth. Clear cell renal cell carcinoma, the most common type of kidney cancer, depends heavily on glutamine metabolism. Although drugs that target glutaminase have been tested, they have only worked in some patients. Another protein, HIF2 alpha, helps these cancer cells survive under stress, but its exact role in metabolism is not fully understood. The biological question of this study is, does HIF2 alpha regulate how glutaminase behaves when glutamine levels are low? The authors hypothesize that HIF2 alpha prevents glutaminase from forming clusters in mitochondria. By limiting this clustering, HIF2 alpha may reduce enzyme activity and help cancer cells in nutrient-poor environments. Assays and methods. First, they used glowing stains and a microscope to see where the enzyme glutaminase was located inside cells. They compared cells with glutaminase to cells without it to see if the enzyme moved or formed groups. Then they removed glutaminase for different amounts of time and added it back in different amounts. This helped them see how quickly the enzyme responded and whether the changes could be reversed. They also watched live cells over time to see what happened to the mitochondria, since that's where the enzyme works. They blocked certain proteins to test whether changes in mitochondria were necessary for the enzyme to cluster. Next, they tracked how active the enzyme was by feeding the cells specially labeled glutamine and measuring how much product was made. They also tested a modified version of the enzyme that naturally forms groups to see if grouping alone increases activity. Finally, they mimicked low oxygen conditions and changed levels of HIF2 alpha to see how it affects enzyme clustering to see if this mattered for cancer growth. They put cancer cells into mice and measured tumor size and cell death. And this is just a restatement of the why, but glutaminase was tracked inside the cells to see how it would respond without proper nutrients. The cells were grown with and without glutaminase and observed for changes. The low oxygen conditions were observed in the mitochondria, and they measured how glutaminase is active and whether grouping changes its activity.
Okay, hi. I'm going to do the results section, so you can go to the next slide, Rishi. Okay, so when the researchers used fluorescent staining to look at glutaminase inside cells, they saw that under normal conditions, the protein appears spread out within the mitochondria. But then when glutamine was removed, glutaminase appeared in small clusters instead. And so you can see these clusters as the bright red dots in the figure. And you can go to the next slide. When glutamine was removed over time, the clusters appeared within about 1 hour looking at the figure and became more noticeable. And when glutamine was added back, the clusters were like no longer observed and glutaminase appeared spread out again inside the mitochondria. And so you can look at that in the green GLS1 signal. You can see how it's like less clustered and it appears more spread out throughout the cell over time. And you can go to the next slide. The researchers also used live imaging to look at mitochondria. So shortly after glutamine was removed, the mitochondria appeared more fragmented, and cells with fragmented mitochondria showed these glutaminase clusters. And when mitochondrial division was blocked, fewer cells showed clustering. And then looking at measuring how active glutaminase is. The researchers measured the glutamate levels because that reflects glutaminase activity. So cells with clustered glutaminase showed higher levels of glutamate over time. And then cells that are expressing this modified version of glutaminase that forms clusters also showed higher glutamate levels compared to cells with the normal enzyme. And you can go to the next slide. And so here, the researchers activated this low oxygen pathway, and under these low oxygen conditions, the glutaminase clusters were not observed when glutamine was removed, and so it was glutamine deprived. And when one specific protein, as mentioned earlier, the HIF2 alpha was removed, these clusters appeared again. And measurements also showed the differences in cell survival under these conditions. And then like finally looking at mice, the researchers looked at tumor growth. So tumors expressing a form of glutaminase that formed clusters grew more slowly and were smaller than tumors expressing the normal form of the protein. And these tumors also showed glutaminase clusters and higher levels of staining for certain cell death markers.
So now that we've gone over the actual findings from the figures, let's briefly review them and also talk about interpretations. So firstly, the key takeaways in the first few figures, the author used various techniques to show that when glutamine levels drop, GLS1 starts clumping inside mitochondria, and it happens quickly and it's also reversible. So we know that this is not damage, but it's rather an active and specifically reversible process, just like flipping a switch. And we also found out that it's not just glutamine itself that matters, but also glutamate, which is a molecule formed from glutamine, as mentioned previously, and prevents clumping when present. And so the interpretation the authors used of the first few figures, was that GLS1 clustering is a warning response when things get risky and nutrients are out of balance. And we personally believe that this interpretation is reasonable because the response is fast, reversible, and it's also tightly tied to levels. Following that, the authors connect enzyme clustering to mitochondrial shape. When the mitochondria break into smaller pieces, GLS1 clustering increases, and when the process is blocked, the clustering goes down. This suggests to the authors that the structure and metabolism are linked and also reversible, and it also helps strengthen the claim that clustering isn't just a chemical effect, but it's more of a whole cellular response. And one of the most important interpretations comes next going down the paper. In figure 5, we find out that when GLS1 clusters, it actually becomes more active. And so, although that sounds helpful at first, we learn that too much activity can overload the cell and push it towards death. So, the interpretation the authors have for this is that GLS1 clustering is a dangerous state for cells under nutrients tests. And this is a really key point, which is that clustering isn't protective and it is harmful. After that, the authors then introduce hypoxia signaling through HIF2 alpha, and they talk about the protein and how it can prevent GLS1 from clustering, specifically to talk about how it doesn't do it by turning genes on or off, but rather physically preventing the clumping. And when HIF2A alpha is removed, the clustering also comes back. And so this directly supports the interpretation that HIF2 alpha helps cancer cells survive by keeping metabolism under control. Then lastly, the mice experiments just show and confirm that what we see in cells also applies to living organisms. And so now we talked about the key takeaways and went through like the meaning of the results. So in terms of major interpretations, there are four major interpretations that we wanted to like focus on. The first one was that HIF2 alpha prevents glutaminase from forming clusters inside mitochondria. The second is preventing clustering keeps glutaminase activity lower and helps cancer cells survive when nutrients are limited. The third one is increased glutaminase activity can become harmful and lead to cell death. And the fourth one is cancer cells control metabolism not only by enzyme amount, but also by enzyme location and organization. So taken together, the authors kind of conclude that CCRCC survives by preventing glutaminase clustering and not by increasing metabolism, but by carefully limiting it. And this allows them to maintain like a careful metabolic balance to avoid nutrient stress. And this also explains why drugs that simply block glutaminase haven't worked very well because the cancer cells are already keeping glutaminase activity low. From our personal perspective, we strongly agree with all the authors' interpretations. Their conclusions are also strongly supported by multiple experiments, consistent results across cell types, and evidence from both lab and animal models.
So that leads to the conclusion. So the authors conclude that the protein HIF2 alpha helps CCRCC, those kind of tumors grow by changing how the protein glucomanase behaves inside the cell's mitochondria. So normally glucomanase can group together or cluster, as was demonstrated in previous slides. And this increases how cells use glutamine, which is an important nutrient. But of course, the study shows that HIF2 alpha prevents this clustering from happening. And by doing this, it reduces glutamine metabolism and protects cancer cells from a type of cell death that can occur when glutamine is processed too actively. So in simple terms, HIF2 alpha helps cancer cells survive by keeping their metabolism at a safer level. This finding is important because it may explain why drugs designed to block glutamase have not worked well in clinical trials for this cancer. So first, things that science could figure out is exactly how HIF2 alpha stops glutamase from clustering. They could also test whether this same mechanism happens in other types of cancer or even normal tissues. And finally, some future studies can include ways to intentionally trigger glutamase clustering so they can selectively kill cancer cells. Thank you.